Welcome, and thank you so much for joining us today for our educational webinar, Clinical Consequences of NIPS No-Calls Methodology Matters. My name is Jacqueline Eng. I am an Associate Director of Clinical Marketing here at Council and will be your moderator for today's event. In this live webinar, we will be exploring the major findings from a recent study published in Prenatal Diagnosis this May, comparing the two main sequencing methods for NIPS. Whole-Genome Sequencing, or WGS, and the Single-Nucleotide Polymorphism, or SNP method. I am pleased and honored to be joined by two authors of the paper, Carrie Haverty and Dr. Dale Muzzy. Carrie Haverty is a Senior Director of Clinical Products for Prenatal Screening at Council. Prior to this, she was a Genetic Counselor Supervisor at Sutter Pacific Medical Foundation and Senior Genetic Counselor at Massachusetts General Hospital. She has her MS from the University of California, Irvine, and is a co-author of the paper we will be reviewing today. Our first speaker will be Dr. Dale Muzzy. He is the Director of Scientific Affairs at Council, as well as a computational biologist and an expert in genomics technology who has led development of high-throughput NGS-based tests for complex disorders, such as spinal muscular atrophy and alpha thalassemia. He received his PhD in biochemistry and biophysics from Harvard University and is a senior author of the paper. We will be accepting questions and comments throughout the webinar and will address as many as possible if we have time. Please use the chat feature to do so. Any that we don't have time for will be forwarded to our speakers post-webinar for follow-up via email. With that, I'll turn it over to our first speaker, Dr. Muzzy.
Thanks, Jacqueline, for the introduction. Thanks to everyone for attending the webinar. We're super excited to be sharing this research with you. I want to start with a holistic view of how prenatal screening has evolved over the decades. It began as a protein-based test for a single disorder that had relatively low sensitivity and has evolved into NIPS today, which is a self-read DNA-based test for many conditions with very high sensitivity. It's this high sensitivity that in the last couple of years has really changed the technique such that it's no longer restricted to high-risk pregnancies, but is now actually recommended for all pregnancies. And this major change in volume means that things that were very rare disturbances in a relatively small volume of patients now becomes actually a potentially like crippling commonality in clinical practice when expanded to all pregnancies. One of these such restrictions is the performance of the test at low fetal fraction. And so we actually looked very deeply at that in a recent paper that we published in Prenatal Diagnosis that compares the WGS and SNP methods of performing NIPS, focusing on their performance at low fetal fraction. So Carrie and I will be walking you through that paper today. I want to start with some of the main conclusions, and then we'll discuss how we reach those conclusions. So first, we've demonstrated that the WGS method has higher sensitivity than the SNP method at low fetal fraction. And secondly, we found that overall, the WGS method leads to fewer no-calls, fewer false negatives, and fewer invasive procedures. I want to focus first on how the two different methods work. And so what I'm showing in this schematic are cell-free DNA fragments. That's what each of these little lines is. You can see that some of them, shown in black, are maternally derived, and the ones in blue are fetal-derived fragments. So on the left, I'm showing a disomic pregnancy, and on the right, a trisomic pregnancy. The difference being that in the trisomic one, you have an excess of fetal DNA. That's those extra 5 fragments at the bottom. This particular schematic is showing a pregnancy where there's 20% fetal fraction. So 20% of the reads are blue and fetal-derived. With the WGS method, the way that it works is we basically sequence in an unbiased manner any of the fragments from the cell-free DNA that we extract from the maternal plasma. And then we map them to the genome and count how many there are in a particular region. So in the region on the left, which is indicated from chromosome 3, we would, if we had sequenced all of those fragments and count them up, there's 50 fragments or 50 reads that we get in a bin. And there are many, many bins across the entire genome and across each chromosome. So you can kind of see that in the top plot where for each chromosome we're plotting many, many different spots, each corresponding to a bin, and all of them have around 50 reads. In the trisomic case, where you have an excess of fetal cell-free DNA, we would actually count 55 reads. And the other bins on chromosome 21 will look very similarly and have that excess of cell-free DNA. So on average, it'll have 55 reads. All the way on the right of the plot, you can see two different distributions, one in red, one in gray. The red one is from chromosome 21, and you can see that it is significantly upward shifted from the gray background distribution. And that is how the WGS method determines whether or not a particular chromosome is trisomic. The SNP method looks at a totally different signal. It does not look at the number of reads per bin. It instead focuses on the allele balance at a whole bunch of SNPs that tile their chromosomes of interest. So let's look at these examples again. At a particular site where you can see those A's and B's, by convention we basically call one allele an A and the other allele a B, and the SNP method focuses on sites where there's known to be high population diversity. And so in many cases, in this one here, the mother will be homozygous for the A allele, and in this case the father would have at least one B allele, such that the fetus could inherit that paternal B allele. And then if you look at the percentage of A at that site, we have 9 A's and 1 B, giving us 90% A fraction, and we plot that below as one red spot at 90%. You can see lots of spots there. That corresponds to a bunch of different SNPs, each of which has the maternal genotype being homozygous for A and the paternal allele giving 1 B. Now, just to orient you to the other spots on the plot, in blue at the bottom would be cases where the maternal genotype is homozygous for B and the paternal genotype gives you an A. And so that basically takes you from a 0% allele balance up to 10%, because you basically have 9 B's and 1 A. The green spots correspond to places where the mother is heterozygous, so she has about a 50-50 mix of A and B, and then the deflections away from 50% represent the paternal allele, either giving you an extra copy of an A or an extra copy of a B. So you can see what those distributions for red, blue, and green look like in a disomic case. Let's now look at a trisomic one, where we'll focus first on that one particular SNP, where you can see that we have a paternally inherited trisomy, such that you have 2 copies now of that B allele, both from dad. And now the allele balance at that site, or the percent of A, is not 90%, it's now 81%. So the entire red distribution of spots shifts downward. Correspondingly, the entire blue distribution of spots shifts upward, and the green distribution dilates. And overall, when you compare the trisomic versus the disomic series of spots, you can see that they're very, very different. And when you can see that by eye, that's basically exactly what the algorithm for the SNP method is also looking at. Now that we've seen how each of those strategies works, I want to really walk through like the methodology of the paper. So we wanted to look at low fetal fraction for any given aneuploidy at hundreds of thousands of affected samples so that we could see how well each of the methods performs. We modeled both positive and negative samples and then saw how well the methods could yield true positive or true negative calls versus how often they yield false positive or false negative calls. And then we finally compiled that all into sensitivity and specificity metrics for each of them. Let's focus on that first part of the workflow, which is testing hundreds of thousands of mock samples, and why we really needed to do mock samples instead of real ones. There literally aren't enough affected pregnancies at very, very low fetal fraction to do a clinical test of hundreds of thousands of them. So you really need to leverage a simulation framework instead. And this is where we get these mock samples. But what's really cool is that we can make a framework where we could actually, create an arbitrary number of these that have any aneuploidy and any fetal fraction in any number. And so we created code that can basically take in as inputs the aneuploidy and the fetal fraction that's desired. It then can simulate the WGS data you would expect and the SNP data that you would expect. And then with those 2 data types generated, we then implemented the algorithms for both the WGS method and the SNP method based on the published and patented information about them, and ran the data through those algorithms to yield calls. And then we could finally assess whether they were true positive, negative, false positive, or false negative, and get sensitivities from those. As I said here, one of the key steps is that you actually simulate the data appropriately. So to show that we were actually doing that correctly, I'll focus here on the SNP method, as it's a bit more complicated to capture. And what you can see on the right is the simulated data that we generated with our code for a 19.2% paternally inherited trisomy 13 sample. And you can see that same data from an experimental data from a SNP method publication in 2014. You can see that there's extremely high correspondence between what we modeled and what was published. And indeed, I would submit that the data is actually cleaner on the right. You can see more distinct banding patterns, and if anything, what that demonstrates is that the data would actually be, in our simulations, would be a best case scenario performance of the SNP method. One of the key things that occurred to us as we were doing this was that there's a very strong dependence of the SNP method on the parent of origin and meiotic stage of origin of the trisomy. So we're going to look in this schematic here at egg development. So this would be a maternally inherited trisomy where the non-disjunction occurs either at meiosis 1 or meiosis 2. In meiosis 1, the two homologous chromosomes missegregate to a single egg precursor, and then that can yield trisomy. In meiosis 2 non-disjunction, the homologous chromosomes segregate appropriately, but the disjunction of the sister chromatids is not appropriate in the second stage of meiosis, and that can also yield a trisomy. What we show at the bottom here now is the SNP data that you would expect from each of these different types of non-disjunction events for a maternal or a paternally inherited trisomy. What I want you to focus on is the difference between the disomic case all the way at the left and each of the trisomic cases. Those are the 4 plots on the right. Now, focus first on the one all the way at the right, the paternal M2 trisomy. This is a huge difference in signal between the trisomic case and the disomic case. You can see a major shift downward of the red spots, a shift upward of the blue spots, and a broadening in the green. This is picked up very, very handily by the algorithm. The thing is, only 7%, as indicated there in parentheses, of trisomies are paternally inherited and M2 non-disjunction caused. What you now want, I now want you to look at the M1 maternal trisomy. This is 70% of trisomies, and you can see that the difference between that M1 maternal trisomy and the disomic case is far more subtle. The red distribution shifts to a very, very small extent. The blue distribution also shifts a tiny bit, and there's almost no dilation in the green. All of these are the same fetal fraction, so this is not a consequence of fetal fraction. This is purely a result of the origin of the trisomy and the meiotic stage of which it occurs. And so the SNP method is highly susceptible to that, and that's really what limits its sensitivity at low fetal fraction. The WGS method, by contrast, is not limited and has no dependence whatsoever on the origin of the trisomy, either maternal or paternal, or the meiotic stage at which it occurs. So that, as you can see, each of those red distributions for the trisomic case look identical. They are the same type of sampling. So the sensitivity of the WGS method is not susceptible to this limitation. So when we put it all together, we were able, as I said, to model hundreds of thousands of pregnancies at any fetal fraction for any chromosomal aneuploidy for the WGS and the SNP methods. And the SNP method really, because of that maternally inherited M1 trisomy limitation, actually has a hit in sensitivity when you go below 4% fetal fraction. At 4% and above, both of the methods work very well on all types of chromosomal aneuploidies. But as you start to get below 4%, the SNP method sensitivity drops off precipitously, whereas the WGS method sensitivity remains high. So again, I think as I've shown you, like, the two methods are very, very different in terms of how they work mechanistically, molecularly, and algorithmically, and also that they have very, very different response to the origin of the trisomy, and that's why you can see this divergence in the methods in their performance at low fetal fraction. The empty red bars, just to be clear, that is the sensitivity expected with the SNP method, but that is also the level at which published reports from SNP method labs indicate that they yield no-call results, which makes sense because the sensitivity there is very low. But you can see even at 2%, there's very high sensitivity for the WGS method across each of the 3 chromosomal aneuploidies. So a common criticism that can be levied against any type of simulation paper is that the parameters used in the simulation may not actually reflect reality. And we are very well aware of that and really built in a lot of checks and balances to make sure that we were doing this in a very careful and transparent way. and made sure that the conclusions we were drawing were robust to any type of parameter change. So what I'm showing here is the effective noise on the SNP method, and we'll do a similar thing to the WGS method in just a second. But what we wanted to see is if you reduced the noise of the SNP method such that it achieved its theoretical minimum, so the cleanest data it could yield, like would the conclusions that we draw still hold? And so as you can see in these 4 plots, as you proceed from left to right, the data get cleaner and cleaner and cleaner, and that Poisson distribution all the way at the right is literally the best that it could theoretically be. So if we now look at the sensitivities of each of these, at that theoretical maximum even, this is now the lower right plot, the WGS method still has higher sensitivity than the SNP method. So there's literally no decrease in noise that could make it better than it is. And so this gives us very high confidence in the validity of our conclusions. For what it's worth, I also want to add that all of the code that we used to generate this, we posted online and made publicly available for everyone to use in the field. So this, in the same way that we wanted to see how well the SNP method could perform at the minimum of noise, we wanted to see the consequences of WGS performance as you add more noise, and to see how susceptible our conclusions were to that. So I'm showing that in this plot where the theoretical minimum of noise is the Poisson panel all the way at the right, and as you add more and more noise, more noise is added as you proceed to the plots toward the left. And you can see that as you would expect in these plots of sensitivity at the bottom, as you add more noise to the WGS method, the sensitivity does fall. But then the question becomes, okay, this is simulated data, so approximately where is the experimental data? And even at the very infancy of the WGS method, when the method was at its coarsest and we had, as a field, very little experience with it, the dispersion observed was actually at a value of about 0.9. So we actually don't have it plotted here, but it's extremely high. It's very much toward the right end of the plot. And what I want to point out is that even at 0.666 dispersion, so this is actually much more dispersed than the data was even observed more than 7 years ago, the WGS method still exceeds the SNP method in terms of performance. And so, it was important to do this analysis, but it's also important to keep in mind experimentally where the data actually, the experimental data suggests that things are. Yeah, so to conclude, each of these, this series of data really demonstrates that the WGS method has higher sensitivity than the SNP method at low fetal fraction in a robust manner, and we were very careful to probe that as deeply as we could. It looks like we have a few questions that are starting to come in. Please do continue to use the chat feature as questions come up, and we will try to address as many of them as possible at the end of the talk. I'd like to now hand it over to Carrie Haverty, who will be reviewing how these differences in methodology translate into clinical impacts for you and your patients.
Okay, thank you, Dale. I'm pleased to be here and share our work. I'm particularly going to talk about the clinical impacts to you and your patients and how we modeled the published clinical experience in our paper. Just some reminders for folks that guidelines are expanding to include average risk patients, and so more screens on more patients means that impacts of things like no calls and test failures is going to have a larger impact on our practice and on our patients. And in fact, we can see here that there are already patients expressing their experience with dealing with no calls. It's just more waiting, anxiety, stress, uncertainty, deciding whether or not to do invasive procedures, waiting for the results of invasive procedures, etc. And I think that we've all talked to patients who are in this sort of state of mind. It's challenging. So one of the things that we did with the paper is that we looked at published clinical experience with non-invasive prenatal screening and test failures to see what happens what would happen in a simulated real world. What do patients actually do with that information, and what is the impact on clinical practice? So we start first with 10,000 idealized samples with low fetal fraction, meaning that these samples all meet every other technical requirement. There's sufficient blood and QC metrics are achieved, etc. And we run those 10,000 samples through either the WGS workflow or the SNP workflow. And with WGS, we'd expect all of those samples to receive a result, take about a week. With the SNP workflow, we know that all of those samples would be a no-call, as defined in the published literature around low fetal fraction samples with the SNP methodology. So all 10,000 of those patients and their supporting providers now need to discuss whether or not to submit a redraw or go on to invasive procedures. Some do choose to do a redraw, of course, and some will have success. And again, these numbers are based on the published clinical experience. Some will choose to do a redraw and not have success, and they will end up with not getting a result, and again having to decide whether or not to do an invasive test. And some will choose not to do a redraw at all and go straight to the decision about whether or not to do invasive testing through amnio or CVS. And of course, ultimately, there will be some who choose not to do an invasive test and then will have no result at all. And of course, all of these things do take time, energy from both the patients and the providers' offices. So just to zoom in here with the SNP method here, we see that ultimately we have 42% of patients, of these 10,000 patients, 42% would receive a requested result after a second blood draw, which takes some time and energy again. 32% would end up getting a diagnostic test, sort of contrary to the goal of NIPS, and the stated reason many patients choose to do NIPS is to avoid an invasive procedure. And ultimately, there will be a number, 26% in this model, that get no results at all. And how many aneuploidies are missed in this group? So we looked at that, overall the numbers, and again, in this group of 10,000 idealized patients with low fetal fraction, we see that WGS ultimately would identify at least 17% more aneuploidies on the first blood draw compared to the SNP methodology, where some percentage in green there would be detected via the second blood draw and some additional percentage would be detected via those who went straight to an invasive diagnostic procedure represented in the blue. Additionally, with no-call samples, there's actually twice as many false negatives because you still have a large percentage of patients, 26% in this group of 10,000 who don't get any result at all. So they neither have a aneuploidy screen from NIPS nor do they have a result from an amnio or CVS, potentially leading to the births of children with aneuploidy without having known about that in advance. And of course, whenever we're doing invasive procedures, while CVS and amnio are very safe, they're not risk-free. There is a small risk of pregnancy loss, and the more procedures you do, the higher the chance of experiencing a pregnancy loss due to an unnecessary invasive procedure. And this just demonstrates exactly how many unnecessary invasive procedures you need to get in order to achieve that stacking of green and blue at the bottom, the detection of those pregnancies identified either by NIPS on a second draw or by an invasive procedure. It's pretty striking. And sort of like Dale described with a model, you want to pressure test the assumptions. In the previous slides, the assumption we were building on was that at most 55% of patients would choose to do an invasive diagnostic test after a no call for NIPS. And again, this is based on published clinical experience literature. But even if we assume it's much lower than that, as low as 5%, you would, of course, have fewer unnecessary invasive procedures and less procedure-induced losses because of those unnecessary procedures. But the trade-off is that you have more false negatives. You have fewer people with information about their pregnancy before birth. And so it's always a constant trade-off with this. So regardless of how high or low we dial that, that option, you can see that there's more unnecessary invasive procedures and more false negatives compared to the WGS method. And this is important, as I said earlier, as we're expanding into the average risk population, our general OB practices, more and more are offering to more and more of their patients. One concern, of course, is that we know that there's an association with higher BMI and lower fetal fraction. And patients with high BMI are a significant portion of our practice, and this is a concern. So this effect of low fetal fraction and having more no calls is going to preferentially hit our higher BMI patients. And as we expand more and more to the general population, our practices will feel that effect. So this was an important question for us to consider and address as we looked at the research to do the paper and a motivation for trying to figure out what really does work best for these low fetal fraction patients. And overall, our outcomes really did demonstrate that WGS has a higher sensitivity than the SNP method at low fetal fraction across all parameters, so as Dale reviewed. And in terms of clinical impact, there are fewer no-calls, fewer false negatives, and fewer unnecessary invasive procedures, leading to potentially, and this wasn't explored in the paper, but a logical conclusion would be less anxiety and less workflow impacts to your practice and your patients. And with that, I'm going to turn it over to Jacqueline because I see that there are some questions for us to review. And thank you so much for your time and attention. We really appreciate the opportunity to go over this important research with you. Thank you.
Okay. Thank you to both of our speakers today. We do see a lot of questions coming in. Please continue to use the chat feature to send these in. We will try to address as many as possible today, but we'll also be able to follow up via email with additional answers. Okay, so the first question that we'll take reads as follows. Given this analysis was based on a simulation, do you have any real-world evidence that confirms these findings? So given the technical nature of this question, Dale, it'd be great to hear a bit more from you on what additional support was used to help support your analysis.
Yeah, thanks for the question. It's a really important one. I just want to start with, like, it is important to do the simulations in a case where you cannot get your hands on patient samples. And in this case, where we really wanted to look at the low end of fetal fraction and across many different trisomies, some of which are rare, there literally would not be enough statistical power to look in real patient samples. That's why we really had to use simulations. So given that constraint, you want to make sure that you do those simulations as carefully as possible and as rigorously as possible. So we were really careful to make sure that what we were simulating matched every bit of the published data that we could get our hands on. And so that included, like, different figure panels, included the methods of all of these papers, diving very deeply into the patents and making sure that we were reproducing what labs that perform the SNP and WGS methods do. Once we had those simulations, it's really important then, and I'd sort of talked about this in the slides as well, to make sure that you pressure test what you've done. So there aren't that many parameters in the simulations, but for instance, for some parameter, let's say that we get the best fit to the experimental data with a value of 10. And it's like, and we can base our conclusions on that value of the parameter being 10, but suppose in real life it's actually 12. Like, it is possible the conclusions would change as a result of that difference between the parameter being 10 or 12. The way that you deal with that in a simulation study is you don't put all eggs in your basket on it being 10. You just say, okay, we think it's 10, but let's look at what let's look at what would happen to the conclusions if you changed it to 1 or 0.1 or 0.001 or from 10 up to 100 or 1,000. And if you can show that your conclusions in the simulation are robust to wild changes in that parameter, we know that the experimental data is somewhere in that parameter space. And if the conclusions hold across that entire parameter space, then I'm actually not that concerned with the precise level at which the experimental data would be. So simulations in that way, when performed correctly and with care, are actually extremely powerful tools.
Okay, thank you, Dale. The second question we'll take reads, I'm not sure I understand your definition of false negatives. Doesn't no calling mitigate the risk of reporting false negatives? So Carrie, can you speak to how the paper defined false negatives and how no calling affects this metric?
Sure, thank you. Yeah, good question. Just to go back, guidelines state that these no-call patients, or these patients that have a no-call result, should get counseling and strongly consider a diagnostic procedure. However, it's really important to consider what happens in these no-call situations. What do patients and providers do with this information or lack thereof? And based on those published clinical experience studies, we know the reality is that many patients will neither get a second blood draw done, nor will they get an invasive diagnostic procedure done. And ultimately, this leads to some of them having a baby born with a chromosome abnormality that was undiagnosed, which is effectively a false negative during the pregnancy. Thanks.
Okay, so our next question that we'll take is, since no one from a SNP lab was involved in this paper, what would be their critique of this analysis? So Dale or Carrie, would either one of you like to take this one?
Yeah, I'll take that question, thanks. So the modeling of co-inheritance of SNPs and haplotypes is a criticism that we've received about our simulations, but there's a bunch of counterpoints to it. First, we're well aware that the SNP method patents do show how to account for interruption of haplotypes due to meiotic recombination events in the parents, and we use these patents when making our simulations. But if you assume an absence of recombination, as we did in those simulations, then the math reduces to an independent treatment of SNPs, which is actually overly favorable to the sensitivity of the SNP method. Next, even if there is an as yet unpublished haplotype handling, and remember, like, all of the work in our paper is actually based on the publications and patents for both methods. There's still another observation that supports our simulations, and let me see if I can find a slide that backs that up. Okay, here. You can see that our data suggests on the left that the no-call threshold for the SNP method should be just below 3% fetal fraction. And on the right, you can see from the published work from SNP labs that the no-call threshold is 2.8% fetal fraction. The close correspondence between what we found with our simulations and what has been published in the literature is really important. If haplotype handling actually boosted sensitivity above what we modeled using independent treatment of SNPs, why would they still have such a cutoff? As long as you assume that the SNP lab is being sensible in their choice of a fetal fraction threshold for no-calls, then if our simulations omitted some sensitivity boosting aspect of their algorithm, that boost would necessarily be offset by one of the many ways in which we gave the SNP method the benefit of the doubt by modeling the data too cleanly. And if you recall, I already showed this data here where our simulated data on the right has much less noise than the published data on the left. And that lower noise level would yield an overestimate of the sensitivity for the SNP method in our simulations relative to the published data. And finally, even if despite all of this, you still thought that the SNP method's as yet undescribed haplotype modeling makes our simulations totally unrealistic, it has no bearing whatsoever on the important conclusions that we have in the last figure of our paper, which reveal the substantial impact on clinics and patients of no-calling all samples below 2.8% fetal fraction.
Thanks, Dale. So our final question that we have time for today will be, how is clinical sensitivity affected? So, for example, mosaicism. So I'll ask Carrie, can you take a crack at that one?
Sure, yeah, great. Yeah, it's a good question. I like hearing that people think about things like clinical sensitivity and analytical sensitivity. So, but from its inception, from the beginning, this paper was really designed to be purely analytical. Since we really wanted to analyze hundreds of thousands of low fetal fraction pregnancies, which would be simply implausible in a true clinical study with human patients. And as we make no pretense of this being real patients, we were not commenting on clinical sensitivity. However, I do want to state that the factors that cause discrepancies between analytical and clinical sensitivity with NIPS, such as confined placental mosaicism or maternal mosaicism, would be expected to affect both the SNP and WGS methods equally, so they would not be expected to make a difference in our study's conclusions. Good question, thank you.
Great, thanks, Carrie. So I do see we have additional questions. Unfortunately, we don't have time to answer any further on this call. However, as I mentioned before, we will be emailing additional responses along with a recording of this webinar. So I did see a couple of people ask if we'll be sharing the slides from this presentation. Again, we will be sending out a recording shortly after today. So if you do have an additional question that you think of after the call, please feel free to email them to the address on the screen, webinar@council.com. And with that, this concludes today's session. So thank you very much for joining. And again, a big thank you to our speakers. Have a wonderful rest of the day.